thank you very much for those nice words, David. And I knew this was a very significant thing, but I wanted to take my time and verify it and learn a little bit more about it before, speaking about it. As I told you before, I actually tried to do this kind of research back in April because I suspected there would be serious overlap between some of the target sequences they're looking for with the PCR test and our human genome because of this confusion about exosomes and what RNA comes from what source and all of this. I looked at a different protocol than the one published here. I looked at one from Germany, and this one is from the Pasteur Institute in France. But it really confirmed my suspicion, and I was glad to see it, that basically one of the primer sequences in the PCR test, according to the Pasteur Institute protocol, is an exact match for a sequence in our own human DNA on chromosome 8. So I thought it might be helpful to take this, and because it's a big thing, it means a lot, but I want it's difficult to kind of understand what are the real implications of a finding like this. So I thought it would be really helpful if we could just understand PCR a little bit better first and just the basics of how it works. So bear with me here. I want to share my screen. And I have a little video here. And this is a mainstream video about the PCR test for COVID-19 specifically. So I'm going to play two different parts here. In the first part, they're talking about where they get the sample from. So listen to this.
The swab of a person with COVID-19 will contain a mixture of human cells, virus particles, and other microbes.
So did you hear that? It will contain a mixture of human cells, virus particles, and other microbes, which could mean bacteria or fungi or other microbes. Okay, so it's not a clean sample. And if we were testing for human DNA or RNA, we could find it because there are human cells in there and also fragments of human cells, such as apoptotic bodies or exosomes, or sometimes there's even free human DNA circulating in some of our fluids. So this is not a pure sample, and it certainly contains human genetic material from the beginning. So if we test for it, we could find it. Now this is another segment of here, and this shows sort of the mechanics of how the test works so you can get an idea.
There's not enough viral RNA to detect directly in the patient's sample, so a process called reverse transcription polymerase chain reaction, RT-PCR, amplifies many copies of a segment of the N gene.
So what they're saying is that there's a tiny, tiny amount of the RNA that we're looking for in this sample that's really messy and contains a lot of stuff. So in order to be able to detect it at such tiny, tiny amounts, they have to amplify it or make multiple copies. And that's really what the PCR test does.
Short, single-stranded pieces of DNA called primers recognize unique RNA sequences within the viral genome that bracket the target region of the N gene. After the first primer binds, an enzyme called reverse transcriptase extends, synthesizes, a single-stranded DNA copy of the viral RNA.
Okay, so I want to stop here because this really shows the important part that we need to pay attention to. That you see in the bottom right hand, there's this short primer, right? So that's the only thing that we have identified in advance. Like, we created the primer. We could make a primer for any sequence, for any organism. And then when we put that in this reaction, basically we're looking for a piece of genetic material from some organism that matches it, is the complement. Okay, because when these two strands go together, they have a complementarity. It's like a plus and a minus that attract each other. And it's G's and C's and A's and T's. Okay, so this is going to look for a specific sequence in the sample that we're trying to find. And what the long strand that you saw in this video is, that's the sequence that could be from another organism that we're identifying. Now, since the primer, at least one of the primers that we have been talking about, is an exact match for a sequence in chromosome 8, then it could identify a piece of a transcript from chromosome 8 rather than some genetic material from another organism. In other words, it can detect our own DNA. So I'll just want to share one more thing here to verify to everyone, because it's really important to make sure that this information is correct. And so this is right here, the sequence of interest, okay, this 18-letter code, and it says it's from the RDRP gene in the virus, okay? Now, I don't know if I can show both of these on the screen at the same time, but I'm going to try my best. So this I'm scrolling through right now is from GenBank, which is part of the National Institutes of Health, where they store all of the sequences from the Human Genome Project and also from other animal genomes. And so you can see Homo sapiens is human, chromosome 8, and then all this gobbledygook here is just basically how they took the cell, all the DNA and split it up for sequencing, and this is the primary assembly when they did it this way. But if you look to the bottom of this, and it gives some references, here's the actual sequence. And that matches exactly this sequence here, which is from, sorry, you're in the way. The, where does it say? The Pasteur Institute, sorry, it's listed on the top in the file name. But this is the Pasteur Institute protocol from the World Health Organization. So you could see those sequences match up perfectly.